Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Hello and welcome to this episode of the Epigenetics Podcast. Today I'm happy to welcome Yadira Soto-Feliciano from MIT on the show. Please let me briefly introduce you to our audience. You got your PhD in 2016 from MIT. You then moved on to do a postdoc at the Rockefeller University and became assistant professor at the MIT Koch Institute for Integrative Cancer Research in 2022.
Right.
A question I like to ask every guest to start off our little podcast is, how did you become interested in biology in the first place and then in pursuing a career in science?
Yes. So for me, the interest and kind of starting as a biologist was a little bit non-traditional. So I actually started as an undergrad in chemical engineering and really interested in understanding chemical processes and how we could apply those to kind of real life problems. But in my quest to become a chemical engineer, I actually learned very quickly when I took my first engineering class that engineering was not something that I particularly enjoyed. And I quickly pivoted into pure chemistry. And this was due because I was doing research in a physical chemistry lab since I was a freshman in college. And it really got me excited about physical sciences and understanding sort of fundamental principles of, how molecules interact with each other. And interestingly enough, the lab was a laser chemistry lab. So I got to play with lasers very early on. And that got me really, really interested in understanding applied chemistry at that level. So I switched into chemistry, and that's where I really got into more fundamental science. But then when I was an undergrad, I was very fortunate to do a summer internship at Stanford where I was doing kind of similar approaches that I was kind of employing back home. So I'm from Puerto Rico, and I did my undergrad at the University of Puerto Rico. And there I understood, how methodology in the sort of field of spectroscopy could be applied to biomedical problems. And at Stanford, I learned that these, seemingly very abstract methods could help us understand pathogenic states in clinical samples. Got me really excited about biomedical science. And there I got interested in biology and biochemistry. So I decided to take a couple years to carry out research after I graduated from college and in a biomedical engineering lab here at MIT. And that got me fully excited to become a biologist. And I was very fortunate to get admitted to the MIT Biology PhD program in 2010, where, the rest is history. So, yeah.
Yeah, let's maybe move on from that, because I want to start in the year 2017. And there was a paper, and I guess this was a paper out of your PhD work in genes and development. There you described the function of the plant homeodomain finger protein 6, PHF6, in leukemia, and defined its role in regulating chromatin accessibility to lineage-specific transcription factors. Can you talk now from maybe 2010 to 2017, how you got into that and about the results of the work?
Yes. So this is, I'm very happy you're bringing up this paper and this story because, again, similar to how I became interested in biology, it was a fairly nonlinear trajectory into that finding and that paper ultimately. So when I started in grad school, coming from chemistry, I thought I was really excited about structural biology and biochemistry. And I thought that's where, the space where I wanted to pursue my PhD studies. However, I decided to look into cancer biology labs, which at the time to me was a completely new field. And I did all of my lab rotations in cancer biology labs. And that's, how I ended up in Michael Heman's lab here at MIT, which was one of the leading labs studying sort of understanding resistance mechanisms between cancer cells and how, when they're exposed to different frontline chemotherapies, we can understand using functional genomics how these mechanisms happen. And in a project that we were studying resistance mechanisms to frontline chemotherapies, we actually kind of turned our attention to more fundamental processes that really keep, in this case, blood cancer cells and their identity sort of fits And these property is really important for cancer cells to exist as a sort of malignant transformed blood cell. And that led us to study this protein known as PHF6, as you mentioned, which is an important chromatin binding factor that is critical for neurodevelopment. And in the context of adult cells, is a little bit less understood. And what we found is that in the context of these B cell malignancies, it is really important to keep the cells looking like a B cell. But then when you perturb the function of this chromatin binding factor, you actually allow the cells to then navigate other potential lineages that the progenitor cell could have taken in the process of development. And basically what we unveil here is a factor that is absolutely critical for maintaining lineage fidelity in a leukemia or blood cancer cell, that then when you remove that factor, you now allow the cell to navigate other developmental trajectories that then basically change the identity of that leukemia and actually making it from a disseminated disease to a more sort of primary tumor-like disease. And that was actually very interesting because it really allow us to demonstrate experimentally at the time a nascent concept known as lineage plasticity and lineage fidelity that now in 2024 is one of the so-called hallmarks of cancer biology. So it was really exciting because we were able to provide, one of the first samples, experimental evidence that this is a concept that is really important for not just maintaining cancer cells, but also that when you perturb these properties, the cells can become something else.
What kind of assays did you use in that study?
So the types of assays, I'm very happy that you're asking this question because you mentioned 2017, right? And 2 years before, across the street at the Broad Institute and in other institutions across the world, we learned about CRISPR. And CRISPR was one of the really important transformative technologies that allow us to make some of the discoveries that we made in that study, because at the time we were limited by the tools that we could utilize to perturb genes in that particular system that we were using. We're mostly using shRNAs that downstream will elicit an RNA interference response that then will lead to downregulation of gene expression. However, that wasn't a sufficiently low level of gene expression for us to unveil the sort of directly associated phenotypes that were part of this PHF6 biology in leukemia cells. So CRISPR was a fundamental sort of gene editing via CRISPR technology that really allowed us to unveil these phenotypic consequences. But then at the time, chip sequencing was relatively, I don't want to say novel, but a new method that was really restricted to a few labs who were really optimizing the method and really making it more accessible to other labs who were not working at the sort of forefront of chromatin biology and epigenetics. So we were very lucky to be across the street from Rick Young's lab at the Whitehead Institute. And with his group, we were able to apply some of the kind of early protocols of chip sequencing to profile the binding sort of modes of PHF6 in the context of these B-cell leukemias, but also other cellular contexts that really allow us to understand the not just chromatin, but also transcriptional targets of this chromatin binding factor that helped us also get some more clarity in terms of the mechanistic sort of underpinnings that, this chromatin factor was regulating in this context. So definitely gene editing by CRISPR was really key. Chip sequencing, absolutely important. And also, in another sort of fortuitous set of events, we were able to apply very early days of attack sequencing, which is one of the methods that we use routinely nowadays to look at chromatin accessibility profiles in cells. So we were able to collaborate with Jason Buenrostro, who at the time just started as a fellow at the Broad. So we were one of the first sort of users of that method that then also allow us to look at that level of chromatin configuration in the context of PHF6 proficiency or PHF6 deficiency in leukemia.
So maybe this is a little bit off topic now, but you talked about the early days of chip, and that chip got optimized for different sample types, targets, whatever it might be. And now we are, I guess, in the same situation with methods like cut and tag, cut and run. Are you also using those? And how does the and if you are, how does the stage now compare to those days in ChIP?
Yeah, definitely. We are potentially late
to the party
users. Yeah, late users of the methods, because I come from the chip sequencing era. So when I did my postdoc, chip sequencing was still the mainstream method to really look at genome-wide chromatin profiling or chromatin binding profiles of transcription factors, histone modifications. So I felt very strongly in favor of this method, that when it's optimized, you shouldn't change it. But then I, like everyone else, started to look more into cut and run, cut and tag as novel methods that will allow us to do things that chip sequencing wouldn't otherwise. And of course, with cut and run, cut and tag, you can decrease the input of cells, which was a major limitation of chip sequencing. Also, the depth of sequencing was also a fairly, I think, substantial advancement that these methods really optimize. And I think it really allows you to look more in depth at, some of the more nuanced changes that might be happening in different experimental conditions. So we are definitely now full on cut and run, cut and tag users. And I have to say, we're very impressed by the scalability, but also the consistency of the method. And I think all of these properties really sum up to something that was really lacking in the era of chip sequencing, which was having a quantitative method to look at changes in the chromatin landscape, but also chromatin binding of transcription factors or other factors for that matter. So I think this is something that I don't want to say underscore or somewhat ignore in the past with chip sequencing, but I think it's something that we are now able to do in a more systematic, sort of reproducible way. So I'm really excited to be one of the current users of these methods, and the data looks amazing. So I'm fully convinced.
Okay, that's good. Good to hear.
Yeah.
So after your PhD, you moved to David Ellis' lab, who unfortunately passed away in January 2023. Can you talk about the decision that made you pick his lab? I mean, it's one of the biggest labs in the world, right?
Yeah. No, this is one of my career transitions that I cherish the most, because, as I said, my PhD started with a more sort of classical cancer biology question that then, because we follow the biology and the science, actually got us to look into the nucleus and chromatin mechanisms, or chromatin-driven mechanisms. And that got me extremely excited about really understand the biochemistry and the molecular biology behind these processes. And of course, Dave's lab was really the top, of the list when I was looking for postdocs. And I was really fortunate to interview in his lab and meet the current members at the time. And I got really excited about joining an environment that was, a set of postdocs and a handful of grad students who were looking at the problem of chromatin biology and epigenetics from different angles. And that was different than, the environment that I was coming from, which was very cancer biology, very sort of focused. But here, the lab was really interesting because you had neuroscientists, immunologists, sort of traditional biochemists, cancer biologists, and, developmental biologists, all coming from different areas of expertise. But the common sort of language to us was our interest in chromatin biology. And I have to say that was extremely transformative to my experience and my training, because I got to learn about all of these other areas of science and biology that I had no clue about. And Dave did such a great job at really kind of keeping that commonality sort of front and center that allowed us to be open-minded, but also benefit from each other's, areas of expertise without having a sense of like competition or kind of stepping on each other's toes. It was more really like a, big group of colleagues and at the end of a family. So it was really special.
A big team that helped each other, right?
Yeah, absolutely.
So out of this postdoc came a paper that was published in 2023 where you discovered a conserved molecular switch between the MLL1 menin and MLL3 for UTX chromatin modifying complexes that dictates response to the Menin-MLL inhibitors. What did you do there, and can you talk about the results?
Yes, absolutely. So this story was really the product of amazing collaborations across the street, inside the lab, and really bridging a number of, in on paper, disparate expertise and I think technologies. And in this particular paper, we were initially interested in using these small molecule inhibitors of the Menin-MLL interaction, protein-protein interaction inhibitors, as a potential kind of compound or tool that will allow us to disrupt this complex and then understand the phenotypic and functional consequences of that biochemical perturbation. And on top of that, we wanted to layer some of my expertise in functional genomics and sort of CRISPR screens to see how understanding at the level of functional perturbations and functional genomics, what are some of the complexes and dependencies that will become apparent in the context of disruption of these important complex. And that was really exciting because at the time when, in the chromatin biology field / cancer epigenetics fields, people used to think about, biochemistry and genetics sort of almost as separate ways of looking at chromatin biology and epigenetic based mechanisms. What we're trying to do here is kind of combining the two in sort of one particular problem. And that was actually quite successful. We unveiled novel, interesting molecular mechanisms that on the textbook were, painted and sort of proposed as very unique properties that these methyltransferase complexes known as MLL, also known as COMPAS, are known to do in cells and importantly during development. So this is a family of enzymes that are known to catalyze the methylation, particularly on lysine 4 of histone H3. And there are 4 different enzymes that basically can form 2 different versions of the complex. A version of the complex that is known to catalyze di and trimethylation of lysine 4 in histone H3 at promoter regions. And that histone modification has been associated with transcriptional activation and also co-transcriptional sort of activation in certain gene promoters. And the enzymes that are known to nucleate that complex are MLL1 and MLL2. And interestingly, in the context of a very specific subset of pediatric leukemias, there's a fraction or sort of a segment of the MLL1 gene that gets fused with one of 80 different fusion partners through chromosomal translocations that then now create an oncogenic fusion protein that doesn't have the methyltransferase activity. However, this new fusion protein is now to create a complex that then can incorporate sort of subunits of the superelongation complex to then assist with fairly efficient co-transcriptional activation. But the Menin-MLL interaction is still preserved in that particular fusion protein. And that's why there's been a lot of excitement to develop these Menin-MLL inhibitors. So that's another layer of complexity that involves MLL1 and MLL2 and also the MLL fusion proteins at promoter regions. But then I mentioned that there's another version of this complex that is formed by the MLL3 or MLL4 methyltransferases that is known to be important for regulating enhancer elements, particularly during development. And this complex is known to catalyze the monomethylation of lysine 4 in histone H3 that is important to then recruit P300 histone acetyltransferase to then activate those cis-actin regulatory elements. And what we found is that, so this is the textbook model. You have the promoter-specific versus the enhancer-specific complexes. And before our study, there was actually no reported or documented evidence that these complexes had any crosstalk, even functionally. So that to us was really exciting because we found a context in which these complexes are almost switching with each other and actually antagonizing each other's kind of air quotes normal functions, because we're talking about a cancer sort of malignant transformed setting in this case. So what we found is that when you disrupt the Menin-MLL complex with these small molecule inhibitors, you actually displace that complex from promoter regions, and that complex falls off, now basically liberating those promoter regions to be occupied by the second enhancer, so-called specific complex. And then that complex comes and co-activates expression of those target genes. And interestingly, the target genes are actually enriched for tumor suppressive functions, basically processes or molecular programs that when things are going wrong will basically either arrest the cycling of a cell, or actually start programmed cell death programs for that particular cell. So then it commits apoptosis. So what we found is that this molecular switch is actually at the center of how these leukemia cells are responding to these menin-MLL inhibitors. And the second layer of complexity to this is that if you're able to perturb the function of the enhancer-specific complex, so-called MLL3/4 complex, you're able to make those leukemia cells completely resistant to these menin-MLL inhibitors. So the clinical implications of this work are fairly major, in my opinion, because you can imagine that a way for, patients who are being tested or clinically tested with these molecules as actually a frontline therapy now, is to acquire mutations in either MLL3, MLL4, or UTX. And that's not a crazy sort of low probability event, particularly because these genes are actually among the most frequently altered genes in cancers that have been documented to date.
Yeah. And this would give you now a new angles to find new ways for those who are resistant to the inhibitors?
Correct. And so we were really excited about these findings because we were able to not just identify fundamental sort of ability of these complexes to antagonize each other's function. And I think that speaks on more fundamental biological mechanisms, but we were able to identify potential sort of acquired resistance mechanisms that cancer cells can acquire. Overexposure of these targeted therapies, but also we sort of identified the different players that can acquire loss of function alterations that then will render these leukemia cells fully resistant to this treatment modality. And this is important because these menin-MLL inhibitors are currently being tested in, I believe, phase 2, almost phase 3 clinical trials. And oftentimes the phase 3 clinical trial is when the patients are now stratified based on their genotypes. And we think that this molecular information can help clinicians to really have a more informed sort of stratification strategy, approach to really determine, okay, these patients are likely not going to respond to these treatment modalities. And in fact, some of the data that we were able to acquire is that if you treat a cell that is efficient for this enhancer-specific complex, you make it grow faster. So you can imagine that you might accelerate the disease in that particular patient even worse if you treat with different modality. So we think that the information is really going to be helpful to the sort of later stages of these clinical trials designs. One additional sort of layer of that study that we were just discussing is that we were able to understand some of the kind of molecular consequences of that switch that I was referring to between the MLL complexes, and trying to understand what is happening downstream to this molecular switch that we can exploit potentially pharmacologically. And one of the interesting transcriptional consequences of this switch is that some of the programs that are basically activated through this process that is induced by the menin-MLL inhibitor is activation of CDK4/6 kinases. And we thought, oh, wait a minute, there's FDA-approved CDK4/6 inhibitors for different types of cancer, but can we use these molecules in combination to see whether we can bypass some of the intrinsic resistance of these leukemia cells that lack UTX, for example? And long and behold, that was actually very exciting to see that even in the context of loss of function of the complex that is important for activating the tumor suppressive network, we can still tap on some of the downstream effects and really have at least some therapeutic effect from these inhibitors. Yeah. So we not only found the problem, but we were also proposing a solution that can help us potentially kind of corner these leukemia cells in a way that they don't have, escape route in a sense.
Yeah, you also found a connection to the CDKN2A tumor suppressor locus, right?
Correct. And that's the sort of connected to this idea of targeting CDK4/6 with palbociclib, for example, which is one of the FDA-approved inhibitors for these cell cycle kinases. And CDKN2C and CDKN2D are actually natural inhibitors of these kinases that are really important for cells to go through the cell cycle quite efficiently. And there's been a number of very elegant studies that have supported the idea that this could be potentially really hopefully effective targets for different types of cancers, including breast cancer and others. And that's why there's been a lot of impetus to develop these molecules, and they're already being used, as frontline therapies in the clinic. So we thought that could be a potentially, I think, effective solution to this molecular problem that we unveiled.
So after your postdoc, you set up your lab at MIT in 2022. So this comes with obviously more responsibilities. You not only are focusing on your own work, but you have to recruit people and things like that. So I guess everything should start running smoothly now, like 2 years later.
Yes.
Yeah.
Yes, absolutely. And interestingly, even though it was 2022, technically a year and a half, almost 2 years after the pandemic started, we were still in the time and era that, everyone was still masking, we were still testing. There was a lot of still kind of social gathering restrictions. But at the same time, I was able to get some students excited and engaged the first year I started here. So I was able to recruit 3 PhD students from the MIT biology graduate program on my first year. So that was really exciting because it wasn't long before I had at least, some lab members who I could bounce off ideas and really get going with some of the experiments. So that was, I think, very fortunate and really helped get the momentum quite rapidly and I think quite effectively. So I've been very fortunate that, for some particular reason, MIT biology students find our research program very exciting and they really want, to be part of it. So I have right now the lab is formed by 5 amazing MIT PhD students, 1 postdoc. I also have 2 research technicians and 1 lab manager and myself. So we're close to 10 lab members now, which it's, pretty exciting.
Yeah, I believe that. So what are the principles you are running your lab by?
Yeah, this is I think a really important question you're asking here, because to me, I think obviously we all come together to my lab. We would, a shared interest in understanding principles of chromatin biology and epigenetic based mechanisms of gene regulation and genome organization. And of course, we have our own way of looking at this problem. We're interested in understanding how complexes assemble on a regulated manner on the genome. But I think that's on the science piece. That's what brings us kind of initially together. But I think on the more cultural and sort of environment and I think philosophy base or aspect, to me, is really, really important and fundamental that every single lab member has, 360 respect for each other. And I think that will carry everything else almost like, by default. So I think that's really important because it really alleviates the idea of, hierarchy or favoritism or any potential issues that might arise that, in a small lab will get amplified even worse, right? And I think this is something that students actually I think it resonates with them a lot because they feel that because it's a relatively new lab, they're really part of building that, environment, culture, and really what our principles are and what we want our day to day to look like. And I'm really honestly super proud of how things have really fell in place with the lab members, because for some reason even if they didn't know each other very well when they were in the first year classmates, they almost, with a few months passing, they become really good friends. And I think it helps a lot with not just more practical things, right? There's, of course, when you join a lab, a lot of training that needs to happen, a lot of showing around where things are at, a lot of, following your older peers to whatever, the next meeting or the next seminar, right? And I think it really helps new members to feel included almost immediately. So, yeah, so I think respect for each other, 360, and it comes, in the form of respecting the space that, all other lab members are going to be, let's say, tissue culture good, right? Make sure that you spend 5 minutes cleaning after yourself so the next lab member doesn't have to spend 5 minutes of your time. And these little things, they add up to, I think, a more substantial, I think, very respectful and I think collegial environment.
So what is it that this team is currently working on?
Yeah, so I alluded to this a little bit in the previous question, but in my lab, we're particularly interested in studying so-called chromatin adapters or scaffolding factors. And to us, this is an area of chromatin biology that has been somewhat understudied for, obvious reasons. It's, biochemically speaking, they're not very tractable factors that are important for regulating chromatin states and also other processes that happen on the chromatin template. So I think historically enzymes have been easier to study because you can biochemically track them. There's quantitative methods that one can use to actually measure perturbations to an enzyme and so on. However, I think the picture is still fairly incomplete, and we have a pretty good idea that these enzymes can be redundant. They can be also, they can have non-enzymatic functions. And I think highlighting that there's a lot of structural activities that are happening that we don't have a clear view. So my lab is particularly interested in understanding how these chromatin adapters or scaffolding proteins are really important for not just nucleating, but also targeting chromatin regulatory complexes like chromatin modifying complexes or chromatin remodeling complexes. So just to give you an example of what these factors are and what they do, and why we're so interested in really tracking their biology. So one of these adapter proteins that I have some experience from my postdoc, and I think there's still a lot of really exciting biology to uncover, is menin. So menin is the adapter protein for the MLL1 and MLL2 complex. However, those are not the only interactions that menin is known to have in cells. And in fact, that's just a subset of complexes that menin is known to nucleate. And interestingly, menin can nucleate a complex that has a co-activating function in cells, or also a co-repressive function in cells. And this is within the same cell type. So for us, it's really interesting to understand how, seemingly simple protein that structurally doesn't have a lot of action. There's no structural domains. There's no structural motifs. It has a fairly long intrinsically disordered region, but not much going on, structurally. And how such a seemingly simple protein can not just target, but also nucleate different flavors of complexes. So we're really interested in understanding the targeting principles that these adapter proteins utilize, either by having inherent ability to sense a chromatin landscape that is permissive or inhibitory to their binding, number one. Also co-opting DNA sequence sort of activities of transcription factors to then guide their targeting. And these two, functions or features are not mutually exclusive. And at the end, we think it's going to be a combination of these mechanisms. That's the first part that we're interested in understanding. Number two, from previous studies, we know that menin is able to mediate protein-protein interactions with a whole repertoire of nuclear proteins. However, not all complexes are made the same. So we want to understand the specificity of recruitment of effector proteins and what is really driving that. Is it the chromatin landscape? Is it conformational change on the adapter protein that might be precluding or including certain protein-protein associations? And number three, what dictates, you have a flavor of complex A versus flavor of complex B, whether A is going to be a complex that is helping transcription versus B is going to be a complex that will preclude transcription. So we're trying to understand that sort of downstream output in gene regulation. And we're now extending that sort of scale and view to not just local gene regulation, but also genome organization. So we have some projects in other adapter proteins trying to understand how potentially they're stabilizing long-range interactions and potentially some of these contacts that are important for maintaining the 3D genome structure in place. And just to layer on top of that, there are disease-associated mutations that actually deviate these functions and potentially exploit certain, pleiotropic activities of these adapter proteins. And we're trying to also really understand what, particularly prevalent disease alleles in cancer and other developmental disorders are, either exploding some of the already there functions or actually expanding some of those activities. So that's kind of the general area that we're particularly interested in.
So for the last 35 minutes, we have been on a journey through your scientific career. Did we miss something important, or would you like to add something?
I think one really exciting thing that I have seen, and from participating in scientific conferences, and smaller gatherings of chromatin biologists and epigenetics sort of interested folks, I think it's really this notion that I think our field is becoming really open and accessible, especially for more junior scientists. And I'm really excited to see that, because I, coming from a different field in cancer biology into chromatin biology and epigenetics, and to some extent into transcription in 2016, 2017, it wasn't as accessible for somebody at the time, an outsider. And I felt that, oh my God, what did I get myself into going to, Cold Spring Harbor meetings? And I have to say, I have seen such a positive change in the right direction in terms of not just including, outsiders, but also I think for more junior trainees, it's becoming more and more open in the sense that we as a field are opening the stage for not just talks from the most prominent scientists all the time, every single meeting, but we're seeing a little bit more diversity in terms of career stages, expertise, and even, really early on trainees. And I'm really excited to see that, because I feel it's going to bring more and more people into our areas, and more people will mean more and more ideas and more sort of ways of thinking about this huge problem that we're facing, and complexity when we look at the genome as a whole, right? So I think that's something that I'm particularly excited about. And, for the trainees out there, just join us. It's becoming more and more accessible and more open. So I think now is the right time.
Yeah, the only thing is if there is enough money, right?
Indeed. And I think one sort of positive byproduct of the pandemic is that a lot of scientific meetings still kept some of their virtual sort of version of the conference still happening. And I think it's really transformative, particularly for international trainees who don't have the funds or the ability to travel overseas for a 3-day conference. I think you can still get some face time with the science that is happening in these really exciting, of course, unbiased fields. But I think that's something that we should not get rid of, because I think it's really going to allow our science to just reach more people in the world. So, but you're absolutely right. Hopefully the funds won't decrease.
That's true. To finish, what is your definition of epigenetics?
Yeah, good. I can get into trouble with this one, but let's go. So to me, I think, of course, there's like 6+ textbook definitions of what epigenetics means. But to me, of course, I come from the chromatin modifications, area of expertise, and I think that can be considered epigenetic. I think changes on our genome that can be translated into phenotypic consequences without changing that primary sequence of DNA. I think that, to me, whether it can be inherited or not, and of course the definition of inheritance, it depends on what temporal scale you're looking at. I think it's something that we have to be a little bit more open-minded. To me, I of course look at epigenetic changes or regulation of gene transcription or genome organization or even DNA damage repair, right? So to me, those changes that are able to, in a regulated manner, recruit complexes that then will have an effect that then will change the phenotype of those cells, I think that, to me, really encompasses what I think are epigenetic mechanisms.
So thank you, Yadira, for your time and for being on the show.
Oh, thank you so much for having me. This is so fun.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening and stay tuned.